process by which the germ layers start to form. So the mesoderm on the ventral portion of the embryo undergoes a process called invagination. It's basically a folding in of the tissue. So here is stage 1, and here in stage 2, the pole cells have migrated up towards the dorsal side, and then you also start to see between stage 2 and stage 3 this kind of folding in, right? So here's the beginning of the invagination fold. Okay, so eventually this mesoderm is going to form a tube that gives rise to muscle and connective tissue. Endoderm starts to give rise to the midgut, and the ectoderm is left as the outer layer of the embryo. So if you look at these different diagrams going from an early embryo to a later embryo that has undergone gastrulation, and you think of this as a fate map, you can trace where the endoderm and these different germ layers originate. So for example, for the mesoderm that's shown in red, we see that if we were to label this with a marker and trace it over time, we can see that the ventral region gives rise to the mesoderm, and that in the later embryo, this mesoderm is inside the body, right? So it's moved from outside all the way inside, it's enclosed, and it's stretched out throughout the embryo. And we'll talk about kind of more of this process in detail later in the semester. Okay, so this is kind of bringing together the movie that I showed you earlier. So this is the ventral furrow. So this is a view from the ventral side. This furrow is the beginning of this folding in, or this invagination. And then later in gastrulation, you have an embryo that looks like this, where this structure, where you start to see the different segments being formed and it's folded around the back, is called the germ band. So the germ band originated here, and it basically extended from the ventral side on top of the dorsal side. Okay, so this is like a very orchestrated, controlled migration and movement of tissues inside the body, around the top, and it has to stop at the right sections of the embryo, and this process is basically laid down by the body patterning genes. Okay, so in the end, you have late gastrulation, you have this embryo where you have a clear dorsal side and a ventral side. And then after segmentation, you start to see very clear divisions between each segment. So you can start to see where the mouth parts will be, the thoracic regions. These are going to be the regions that have the wings later on. And then the abdominal regions. These will have the legs. And we'll see how changing the body patterning genes in these segments can alter the adult body plan. So now you can go back to this movie with this information in mind and try to think what's happening as the embryo is undergoing this gastrulation and these changes. So the parallel genes, as I said, define the 14 parasegments, and they activate the segmentation genes. Okay, and so the segmentation genes are also called segment polarity genes. And again, this is all happening between the syncytial blastoderm and cellular blastoderm stages, where the segment polarity genes are starting to be expressed. So again, gap genes turn on parallel genes control segment polarity genes, and the segment polarity genes help define the segments. Okay, so let's go back to our gene expression. So this is an embryo at the germ band extension stage. This is immunohistochemical staining for a gene called engrailed, which is a segment polarity gene. And we can see that it's expressed again in very specific regions. And when they looked at the individual cells in each parasegment, it was seen that the engrailed protein is expressed only in the anterior portion of each parasegment. Okay, so it's hard to tell here. Okay, so in this diagram, you kind of see the lateral view of an embryo where the parallel genes are starting to be expressed in alternate stripes. And so you see alternating colors of blue and purple, right, for Eve and Fushi Taratsu. But the boundaries between each of these stripes are still kind of fuzzy at this point. At the stage of development where engrailed starts to be expressed, it's expressed only in the anterior cells of each parasegment. So engrailed is shown here. It's basically the cells that have the purple circles in them. So engrailed expression is really specifically expressed only in the anterior cells of the parasegment. And this starts to help define where the boundaries will be between each future segment. So the segment polarity genes are named because they provide polarity to, or they provide a sideness to each parasegment. It tells which cell is the anterior side and which cell is the posterior side of each segment. And so after this stage, you start to get more defined boundaries. Okay, so if we look at expression of more segmentation genes, such as hedgehog, wingless, and patched, and we look at their expression in the parasegments and compare their expression in the corresponding segments, we can see different patterns. So, like I told you, engrailed is only expressed, and hedgehog too, are only expressed in the anterior cell of each parasegment. So this corresponds to the posteriormost cell of each segment. So brown color represents expression, white is no expression. Okay, so in contrast, there's another segment polarity gene called wingless, and this gene is only expressed in the posteriormost cell of the parasegment. And then there's another set of segment polarity genes called patched, which are expressed in most of the posterior cells of the parasegment, which corresponds to the anteriormost cells of the segment. Okay. So how do these genes work together to kind of define these different segment boundaries? So we have to think, we can see how they interact. So if we look at an individual parasegment, so if you look at the boundary, okay, between two parasegments, we can see here that engrailed and hedgehog are expressed at the more anterior regions of the parasegment, like shown here. And wingless is expressed in the most posterior cell of the previous parasegment, so its neighbor. Okay, so we talked about positive and negative feedback loops in previous classes. And so it turns out that the wingless and the hedgehog and engrailed genes work in positive feedback loops. Their hedgehog and wingless are secreted proteins. They can bind to receptors on these cells, on their neighbors, and they will